Yes.
Yes.
Can you hear me?
Yes, we can hear
Somebody
you.
say something. Can you hear me? Robert, can you hear me?
Yes, Frank, I can hear you.
Let's see. Jesus Christ. Oh, yes, we can hear you. Okay, people do it in the chat. All right. Hey, so thank you for signing up for this exciting colloquium. I see 12 people here, but including myself, but I think we are actually more, at least 15 or 16 signed up officially, and I know that some may join without having officially signed up, which is great. I'm actually very happy that I was able to organize this colloquium. So I managed basically to bring excellent research labs together around the Munich area to present their work on epigenetics. And you may not know this, but Munich is a hotspot for epigenetic research, mainly in the mammalian field, but offers a variety of angles into this very complex field. And I think it's a great opportunity for anyone listening to get an overview of what's going on. Okay, so I have the pleasure of the introductory lecture, and I'm going to keep it quite brief. I want to talk about epigenetics. I want to define it so that we're all on the same page to understand what kind of phenomena we're looking at. So I'm going to start with a joke. A lot of people in the epigenetics field use this joke, and it says, if they ask you anything you don't know, just say it's due to epigenetics. I don't know if you're laughing. It's maybe funny, maybe not. So the question is, what is the joke? And the joke basically implies that epigenetics captures a set of unexplainable phenomena, and everybody should know that, and that's why it's funny. And maybe that's funny, maybe not. It certainly had some truth value to it maybe 30 years ago or 20 years ago. But epigenetics as a field has made some major advances to actually define and measure and model the kinds of phenomena they're actually interested in. So from a modern perspective, actually, this is not a very appropriate joke anymore. And hopefully in the course of this colloquium series, you will see why. One of the problems is that misconceptions about epigenetics have made it into the popular press and dominate some of the headlines. You can see this right here on a Time magazine from 2010, Why Your DNA Isn't Your Destiny. Here on a German magazine, Der Spiegel, essentially says smarter, healthier, happier, how you can trick your heredity. And there have been lots of headline news about epigenetics in the popular press and in popular science magazines like The Scientist. There have been books about epigenetics and how it may bring about a new revolution in biology, as captured in this book right here, or a book on epigenetics, the death of the genetic theory of disease transmission. Some books or magazines even claim that epigenetics brings with it the death of Darwin, or Darwin was wrong. Or more historically bent books like this one right here, which is called Lysenko's Ghost, which makes a case that modern epigenetic research is resurrecting Lysenko's ghost. And if we can just maybe take one person who sort of gave epigenetics a bad reputation, it's certainly this Trofim Lysenko, Very briefly, who was this guy? So he was a Soviet agronomist and botanist. He lived in a time where um, genetics was rapidly establishing itself as a field, but he very early on rejected Mendelian genetics in favor of Lamarckism. To him, the environment defined the essence of organisms, which was very consistent with the communist philosophy, namely that the essence is not a property of individuals themselves, but is bestowed on individuals, in this case by the environment. He believed that acquired somatic changes can be transmitted to the egg and determine the next generation. The problem with all of this was that he mixed communist ideology or philosophy with scientific theory and hypothesis testing. His theories were translated into agricultural policy, and his practices had devastating impact on Soviet agriculture that led to numerous famines, also later in China. So he was a plant person, and some of his unsubstantiated claims were: one, vernalization is heritable offspring. Vernalization is a process where cold treatment of seeds is necessary for plants to flower later on. We now know that this kind of signal is just restricted to a single generation, is not heritable. And I didn't believe that. He believed that crafted scions improved rootstock, meaning that the information flow is from scion to rootstock, but we know it's not the case. He believed that planting seeds close together improves yield because, quote, seeds of the same class don't compete. So, so he certainly gave epigenetics a bad reputation, and there's efforts in the scientific community, like this one right here, Nature Reviews Genetics in 2013, to try to set the record straight. Here we see written many of the recent front page headlines make unsubstantiated claims that are not necessarily supported by current empirical research. So I think, given this confusion often that you see in the popular literature, or even among the laymen, it's important to try to define epigenetics for ourselves, get a clear picture of what we're talking about. And I would like to do this here, giving you a little bit of a historical perspective, which is just supposed to give you a taste of where this definition is coming from that we're currently using. So typically, historians of science attribute the definition of epigenetics to a dual origin, namely Conrad Wannington and David Nanny. But I also would like to mention Joshua Lederberg and Robin Holliday in this historical perspective. So first, Conrad Waddington, he lived from, I cannot see this because I have this stupid thing in here on the top of the screen, but I think it's 1905 to 1973 or so. So he was by training embryologist and developmental biologist, but he also had a very strong interest in evolutionary biology. In fact, he was one member of a group of people who tried to forge the new modern synthesis in evolutionary biology, which tried to bring together the school of biometricians who studied essentially the genetics of quantitative characters, and the school of Mendelian genetics, which studied the inheritance of discrete or categorical characters. And the synthesis was mainly in the form of population genetic theory and mathematical models, mainly put together by Fisher, Haldane, and Sewall-Wright. But he criticized some of the sort of modeling assumptions in modern synthesis. One of the things that the modern synthesis, these new evolutionary models, completely neglected is the development of the individual self. And Conrad Waddington believed that this is an important part that should somehow be brought back into evolutionary theory, because he believed that the things that happen to an individual during its lifetime somehow need to feed back to evolutionary processes. Now, we had no good understanding how this could be, but now there's a whole field that studies precisely this question, which is evo-devo. Nonetheless, so he had an interest in evolutionary biology, but by training was a developmental biologist. He's the one that coined the term epigenetics in 1942. And what he did is, so you need to understand these people a little bit in the context of their scientific environment. So Conrad Waddington still lived in a time where there was a debate among two schools of thought on development. These are these people believing in preformationism and the ones that believed in epigenesis. Preformationism was the idea, the theory that development, the process of development basically makes a smaller organism that is already complete in its entirety found in the sex cell larger. So the whole process of development is just making things that are already fully differentiated just larger. I mean, from a modern perspective, this is a silly notion, but given the limitation of microscopy, for instance, I mean, this was not a hypothesis to easily reject. The other school of thought that we're more familiar with now is epigenesis, and the idea is that the embryo gradually emerges from a formless egg. But for Waddington, these two views were conceptually compatible. To him, the part of preformationism represented the static nature of the gene or the genotype, whereas the epigenesis part represents the dynamic nature of gene expression. So keeping this in mind, the definition of epigenetics that he gave is that epigenetics is a branch of biology which studies the causal interactions between genes and their products or proteins, which bring the phenotype into being. So he did that by way of a very powerful metaphor, which is now known as the Waddington epigenetic landscape, which a lot of people are still using in their presentations now. You can see a picture of this right here. This is from his 1942 paper. And there he basically writes, this is a part of an epigenetic landscape. The path followed by the ball as it rolls down towards the spectator corresponds to the developmental history of a particular part of the egg. There's first an alternative towards the right or the left. Along the former path, a second alternative is offered along the path to left. The main channel continues leftward, but there's an alternative path, which, however, can only be reached over a threshold, okay? So from a modern perspective, it's very easy now to reinterpret the Waddington landscape in the context of cellular differentiation. You start out with pluripotent cells that in the course of development need to make some differentiation decisions and eventually differentiate into terminal cell types, like blood cells or muscle cells, nerve cells or sperm cells. One thing that may not be so easy to understand is what the different dimensions of this landscape actually mean. So I just want to maybe discuss that here for a second. So I mean, there are three dimensions, and some of them are easy to decipher. This dimension obviously is time. It's developmental time measured maybe in cell divisions. This dimension, people who work with these landscapes, modeling at least, interpret this as output, and they interpret this as the total output of all genes and their products arising from their interactions. And here is only one dimension shown, but this needs to be interpreted as a multidimensional space. So for instance, we can think of this maybe as an expression output of gene A. But if, you know, for a given expression value, you can see in this landscape, you can have different values on this dimension. So to make the differentiated cells, the contribution of this gene to this differentiated cell clear, this has to be a multidimensional picture involving more than just one gene that determine a point in this landscape. So this dimension is a little bit more difficult to think about, and in fact, they call this the potential. The potential is defined as the function whose derivative yields the speed at which the ball moves towards its steady state. In other words, how much energy would it take to move the ball away from a point in the surface? I've drawn 3 points in here. You can see quite clearly, here we've got a point here on this ridge. It certainly doesn't take much energy to move it away from this ridge into either one of the valleys. Maybe it will just roll by itself. So here we've got a ball that's already moving. You probably have to do nothing. It just rolls down and then finds a steady state in this valley. And here we've got another point that's already we had a kind of steady state, and it would take a lot of energy to get this guy out of there. The nice thing about so this landscape brings with it two important concepts. One is the concept of plasticity. And in the context of this landscape, it means that the same genotype can give rise to different cellular phenotypes. Right, we're looking at differentiation of cells right here that happen in an organism with a given genotype, but nonetheless, you can mount different cellular phenotypes over time. So there's a plasticity aspect, but there's also another concept called canalization or robustness. And what this means is that the developmental trajectories for some cellular phenotypes are not easily perturbed. So we all, all of us develop normally. We typically develop 2 arms and fully differentiated hair cells and heart cells and so on and so forth. And this process of differentiation is highly canalized. It's very difficult to perturb it. And once you're fully differentiated, it's also very difficult to change the functional state of these cells and differentiate it into another cell type. And equally, it's very difficult to reverse time going from a differentiated cell to a pluripotent stem cell state. Nonetheless, both of these concepts imply a decoupling between genotype and phenotype. And this is a very important aspect when we think about epigenetics, that there is a decoupling between genotype and phenotype. So there's certainly some things now in our arsenal as scientists where we can challenge Waddington's epigenetic landscape, experimentally at least. So you have this picture again under normal, in normal development. But we now know that reversing developmental histories is possible, for example, through cloning in iPS, with iPS cells, as shown right here. Direct conversion is possible from one cell type to another. But this requires a lot of energy or severe perturbations of global gene expression programs. Okay, but we have to overcome somehow this strong canalization that is there. So the question is always, who would Conrad Waddington be in modern terms? I would maybe argue that he's probably interested in gene expression, network dynamics during cellular differentiation. Unfortunately, he wasn't really a molecular biologist, so he made very little reference to the physical components that establish and maintain gene expression states. Another person around this time, we became a little bit later, is David Nanny. He was more of a molecular biologist, but also working in developmental biology, mostly with protozoa, unicellular organisms. And in a lot of his experiments, he observed phenotypic variation in clonal lineages. I mean, so lineages of these unicellular organisms that were genetically identical, but nonetheless showed phenotypic differentiation, would seem to be stable through propagation of these organisms. And he tried to get a little bit more at the molecular mechanisms that maintain different alternative functional cellular states. And he came up with a definition of epigenetics that reads: cells with the same genotypes or genes may not only manifest different phenotypes, but these differences in expressed potentialities may persist indefinitely during cellular division in essentially the same environment. So David Nanny was more interested in the molecular components that facilitate this inheritance of these cellular states across cell divisions. And again, so he was a bit a victim of his time. There was a debate at the time still on what the nature of these cellular inheritances are. There was the school of chromosomal inheritance and the school of extrachromosomal inheritance. So for instance, at the time, it was not entirely clear what the molecular causes for cellular differentiation are. The chromosomal people thought that you actually alter your chromosomes. They physically change, and one interpretation of this is that you could lose DNA, for instance, yeah, permanently. And the alternative is that there are some molecular components that are not attached to chromosomes, they're independent of chromosomes that facilitate the cellular inheritance or the cellular differentiation. I always ask, I mean, now we know that this is maybe a bit of a silly hypothesis to say that maybe, you know, there's the cellular differentiation in a human being is due to genetic changes that occur during development. Like I've said, losing part of your chromosome or something like that. But the question is, how would you disprove this hypothesis with modern techniques? And it's maybe not that straightforward. So one common answer is sequence. You just you know, sequence the genomes of different cell types, and then you can, if you don't see any changes, then obviously you can rule this out. But you know, this is not. First of all, it's costly, and second of all, it's not straightforward for people who work in genome analysis to detect these differences, particularly if they're very small and rare. But a more beautiful experimental approach was provided by, and you probably know it, by John Gordon, who pioneered essentially nuclear transfer cloning. So what he had done is he basically took the nuclei of somatic cells of a frog and transplanted them or inserted them in an enucleated egg of a frog, and got back a tadpole, a living tadpole. So basically, this demonstrated that it cannot be that you're altering your DNA, because otherwise you wouldn't get back a tadpole. So it had to be something extra chromosomal, so to say. So David Nanny championed the idea of, and this now begins to sound very esoteric, and I personally don't really know exactly what he means by that. Epigenetic control systems, he viewed that as self-perpetuating steady functional states organized by cytoplasmic components. One can maybe think of this as sort of metabolic feed-forward loops that are established in the cytoplasm and then somehow maintain themselves through cell division. So once they're set up, they kind of maintain themselves this way. Regardless of what it is, what he believed that epigenetic changes should not result in a permanent loss of information, and a return to a previous condition of expression is always theoretical possible. So this idea that you can have a reversion of an epigenetic change is an essential aspect of epigenetic research. I want to mention Joshua Lederberg because I think he made a very nice conceptual distinction that nobody really talks about much, maybe because Joshua Lederberg went to the wrong parties at the time, but certainly his terminology was not catching on. But from a modern perspective, this is quite right on the point. So he basically says that chromosome inheritance, in the way that I mentioned it, that you need to have physical changes of chromosomes, does not necessarily imply genetic inheritance. I wanted to make that clear. And to make that distinction, he differentiate between nucleic inheritance, and that is referring to genetic information in the form of DNA sequence nucleotides, and epinucleic inheritance, referring to nucleic acid configurations or chemical modifications. And in fact, when we talk about epigenetics now, we're talking about molecular components that are essentially epinucleic. They refer to nucleic acid configurations and chemical modification. And then he made the third distinction is this extranucleic, referring to molecules or reaction cycles not directly connected with nucleic acid. And this is probably something that David Nanny was after. And David Nanny, along with his peers, kind of lumped chromosomal inheritance together with genetic inheritance. So this distinction here was quite neat. So nonetheless, there was very little knowledge of what these epinucleic components could be at the molecular level at the time. So one of we're beginning to get at the modern definition of epigenetics. This one came with Robin Holliday, and he's known for the Holliday junction. He studied DNA methylation in cancer cell lines. He described DNA methylation-based mechanisms for stable cellular inheritance and developmental switching of gene expression states. So he proposed a two-part definition in 1984. And the first part of the definition is the study of changes in gene expression which occur in organisms with differentiated cells and the mitotic inheritance of given patterns of gene expression. Okay, and so basically, he's interested in this aspect, the Waddington landscape we talked about. Gene expression changes have to be established at these branch points where differentiation decisions are made. But once you're fully differentiated, you have to make sure that you maintain these gene expression states through mitotic cell divisions. But for whatever, I don't know what he was on at the time, but he proposed the second part of the definition that was really based on essentially no empirical evidence or very little, no real empirical evidence at the time. And that is, it's also the study of the transmission of information from one generation to the next, other than the DNA sequence itself. In other words, nuclear inheritance, which is not based on differences in DNA sequence. So if we take the second part of the definition and try to project this onto the Waddington landscape, so what he really means to say is that somehow during the differentiation of the sex cells, changes can occur through whatever process that change essentially the functional state of these sex cells or the transcriptome of these sex cells, and these changes then meiotically inherited. So the consequences of Holliday's dual definition, as what it really implies, is that you get this meiotic inheritance through this change, and in the next generation, somehow the changes that are required in the sex cells condition the initial state of the pluripotent stem cells and lead to slightly different developmental trajectories. Yeah. And then there may be an additional round of meiotic inheritance, who knows. But so you had this two-part definition, which at the time was based, the second part was based on very little empirical evidence, but this kind of dual definition kind of stuck around. Maybe partly because later people that built on this definition, like the one from Riggs in 1996, already had some empirical evidence, notably from plants, that did show that epigenetic changes can be inherited across generations. So here's a definition by, a more modern definition by Riggs from 1996. Study of mitotically and or meiotically heritable changes in gene function that cannot be explained by changes in the DNA sequence. So that we maybe think that's a good definition. You can maybe work with that. Personally, I don't like it too much for two reasons. One is that heritable changes is not really defined. So, you're not really making any reference to molecular components that could bring about this heritable change. So when we talk about a heritable change in this definition, it might as well be any type of molecule metabolite that affects gene expression, and that is inherited through the sex cells to the next generation. Yeah. Another one is that epigenetic inheritance needs to affect gene function. Okay, you could argue we could also study epigenetic inheritance that is functionally neutral. It's still interesting to look at the molecular basis of these forms of inheritance without there being any genetic change or phenotypic change. So personally, I prefer a later definition, 2008, which is the study of stably heritable phenotypes resulting from changes in a chromosome without alterations in a DNA sequence. I would just so this is a nice definition, because now we're talking about changes in a chromosome. So we're talking about now epigenetic components that I would just highlight in the next few slides that have physical locations on chromosomes. So we can actually study them and observe them going through generations if we want to. This makes the definition much more crisp. But I would just suggest two modifications to this definition. I would put in parentheses stable, because who knows what stable means? What frequency of change are we talking about that we don't call something stable anymore? So I would put this in parentheses, and again, heritable phenotypes resulting from, again, if these epigenetic changes have no phenotypic consequences, they are still, in my view, considered epigenetic inheritance or fall in the umbrella of epigenetics, but it just don't have any phenotypic consequences. That's fine. It's still interesting to study them. So the key questions we're trying to get at in this colloquium is essentially what changes in chromosomes are we actually talking about here at the molecular level, in this definition? How can they be measured and analyzed in a genome-wide fashion? How are they inherited across mitotic and meiotic cell divisions? And how does epigenetics contribute? Sorry, this should be organismal development and organism-environment interactions, phenotypic inheritance, and evolution, if at all. And you can see these kinds of questions move from perhaps least complex to more complex. I mean, certain straddle scales of analysis. They go from molecular components to possibly studies of ecosystem or functional evolutionary processes. So in this colloquium, we put together I put together at least some groups that would chip away at some of these questions, and I tried to order them in such a way to reflect the order of the question. It was not always possible because of people's schedules, but what we have here for this semester is my introduction, and I'm just giving right now. Then we have something on measuring and analyzing epigenomes from Maria Colometache, measuring and analyzing epigenomes by the Bonev lab, measuring and analyzing epigenomes, the Heining lab, epigenetics and development, the Stricker lab, something on epigenetics and disease from the Siegel lab, epigenetics and aging from the lab of Binder lab at the Max Planck Institute, epigenetics and disease from the Uhlenhaut lab, epigenetics and nutrition from the De Angelis lab, epigenetics and nutrition from the Teperino lab. I'm going to talk about epigenetic inheritance in plants. Then we have the Becker lab talking about epigenetics in a kind of ecological setting, and we're going to finish off with some evidence of a role in epigenetics in evolution. And so with that, I think we're kind of working our way through these questions. But very briefly, before we move into next week's lecture, I'm just going way too fast today. So before we go into the next week's lecture, we just want to very briefly talk about this first question: what changes in chromosomes are we talking about at a molecular level? We all know, you probably all have a little bit of background in epigenetics. We know that DNA is tightly packaged in cells, and there are different levels of organization. The most basic unit of organization is the nucleosome, which has about 147 base pairs of DNA wrapped around it. The nucleosomes are comprised of histone proteins whose amino acid tails can be chemically modified. We can have different histone variant proteins substituted into the nucleosomes. We can modify DNA directly through, for example, cytosine methylation. And we can have non-coding RNAs that can also alter essentially the organization of genomes. So non-coding RNA is always a bit of a funny thing because, as I mentioned, they don't really fulfill the requirement of being able to be mapped to a chromosome per se, and not really stable features of chromosomes floating around in the nucleus, the cytoplasm. But nonetheless, they have a central role in at least in the establishment of DNA methylation patterns and in altering chromatin structure. So one can include them as a molecular component of the epigenome. So anyway, what we typically refer to as chromatin is really DNA plus histones plus RNA. Okay. So the functional properties of chromatin can be altered in several ways. We can change post-translational modifications of these histone proteins right here, or DNA methylation. We can replace canonical histones that comprise these nucleosomes with more specialized histone variant types. We can move histone cores around along the DNA. We can partially or completely remove histones from the DNA. There can also be higher order topological changes that are not really reflected here in the way I show the DNA, but at higher level of packaging, there can be 3D topological changes as well. So we're talking about all of these aspects in the coming days, in the coming weeks. So very briefly, this is more in a cartoon type of fashion. Now we know that epigenetics defines the activity status of DNA, and I've shown this here in the schematic. So here you have what is called closed chromatin, where DNA is a sort of compact, condensed state. And transcription in this region is typically off for the reason that regulatory proteins or transcription factors are unable to physically reach the DNA to start transcription. These types of closed chromatin regions are seen in heterochromatic parts of the DNA. They're defined by repressive histone modifications, often plus dense DNA methylation. We can also have regions of open chromatin, which are typically located in euchromatin. And there you have basically activating histone modifications, as well as a lack of DNA methylation. And these chemical properties open up the DNA, which now becomes accessible to transcription factors, polymerases, and turn on transcription, basically. So one of the interesting things is that we need to think of a combination of specific epigenetic modifications that define a chromatin state. When you look at any given position in the genome, there's always a co-occurrence of different histone modifications, DNA methylation. And it's really the combination of all of those that define a chromatin state, which in turn defines if the DNA is active or repressed, or how active it is, or how repressed it is. So in fact, we should not just think in this on and off kind of fashion as shown here, but also think of it in more quantitative fashion. So a chromatin state can regulate transcription intensity in a quantitative fashion. So these types of closed and open chromatin patterns, they can be seen on a small or large genomic regions. For instance, they can be quite localized in transcription factor binding sites, or they can occur over long stretches, for example, repetitive DNA. So, these chromatin states need to be established and maintained. The establishment means that you kind of move from a, say, open chromatin configuration to a closed chromatin configuration. So what has to happen there? There have to be molecular processes that facilitate this. Some of them include histone deacetylation. So you want to get rid of acetylation states of these histone modifications. Depending on what kind of histone methylation you have, either or demethylated or methylate certain amino acids on the histone tails. You establish DNA methylation as you move from the open chromatin to closed chromatin. Vice versa, moving from a closed chromatin configuration to open chromatin, you have to acetylate your histone amino acid tails. You have to demethylate or methylate certain amino acids, depending on which ones they are. You have to probably demethylate the DNA, and you have to introduce some transcription factors. We're talking about some, if we're talking about genic regions. So this, this going from open chromatin to closed stream is the process of establishing this. And so this is a dynamic and reversible process. And this can be triggered by environmental cues or developmental cues, or sometimes even by DNA mutations. So when we then look at the maintenance part, and this is now the part where, so the establishment part is, by the way, also in Waddington's landscape, the kind of stuff that needs to happen at these branch points. When you make a differentiation decision, some of this has to happen. Yeah. But once cellular functional state is established, the underlying chromatin states have to be maintained over further mitotic and sometimes even meiotic cell divisions. So once you're in a closed chromatin state in a differentiated cell, you need to mitotically maintain this. Okay, so when it's off, it stays off. Similarly, if you're in an open chromatin state, you need to maintain this. So you need to ensure, at this point, you need to ensure stability. So the chromatin state needs to be heritable across mitotic cell divisions, sometimes also across meiotic cell divisions. That means across generations. And clearly there needs to be mechanisms that establish or copy the chromatin states into newly replicated cells without error. So hopefully we hear something about that from someone. I'm not so sure, but these mechanisms exist. Okay. But there are mistakes that can occur in the maintenance of these chromatin states. So you are at one time point, you're in open chromatin and you want to maintain it. By some error, you end up in a closed chromatin state. Similarly, you could start with a closed chromatin state. You're supposed to maintain it, but you make some mistakes. Now you're in an open chromatin configuration. These types of maintenance mistakes can have phenotypic consequences, and they're sometimes observed in cancers, for instance. So in a lot of the talks that we will probably see in the coming weeks, we will see aspects of these molecular processes. And what is clear is that these processes, the establishment of chromatin states or the maintenance of chromatin states, can be influenced by genetics or the environment. And these chromatin states then can have changes can have an impact on development and disease. So in this framework, I would like to just present the talks that you will see in the coming weeks. So I've gone way too fast. I should have done this in an hour and a half, but I sort of flew through it. Are there any questions? I mean, if there are no questions, then I would just have to think. Do you guys want to see the slides? Do you want them posted somewhere, or? Let's see. Maybe there's nothing in the chat. Okay. Yeah, that would be nice. Okay, I will try to see if I can post this somehow on Moodle. Well, this might actually be problematic now that I think about it, especially if people have no access to our Moodle. Well, what I will do is maybe I'll just, if you send me your emails, I'll make an email list, and I'll just send you the slides, and I will ask the other presenters as well to send you the slides. Okay. So if there are no more questions, then I'm really looking forward to next week, where we're really talking about where things are a little bit more detailed and less conceptual. Okay, then see you next time. Have a good day.